عزيز، يعني احنا حقيقة من أكبر الثورات اللي موجودة في العالم المعاصر هي ثورة الجينات. واتسون وكريك وضعوا الدي ان اي كمركب أولي. لو رجعوا اليوم وشافوا ما يحدث الآن على مستوى الدي ان اي في العالم والتطبيقات اللي قاعدة تشتغل في الدي ان اي في العالم، سيبهروا بهذا الإنجاز العظيم وما تم تطويره في العالم خلال الحقبة التي قد لا تتجاوز 50 سنة ماضية. واليوم بشرفنا أن يكون معنا الدكتور صلاح الدين زايد. الدكتور صلاح سيتحدث عن تصنيف التمور وعن طريق البصمة الوراثية باستخدام إحدى الطرق الجينية المستخدمة في هذا المجال. والدكتور صلاح سيتحدث معنا لأنه الدكتور محمد مجدلاوي اعتذر أن يكون معنا اليوم، فالدكتور صلاح راح يتحدث لنا في هذا الموضوع الهام. دكتور صلاح تفضل.
Good morning. I will be presenting in English today for those of you who require translation. I'd like to thank the Ministry of Agriculture, the Jordanian people, and the Khalifa Award for Date Palm and Agriculture Innovation for giving us this opportunity to present our research today. The presentation I will be presenting is titled Date Palm Cultivar Identification Through Simple Sequence Repeats, SSRs, a practical molecular approach for next generation commercial date palm micropropagation and general Palmàprinting or fingerprinting. This is a beautiful orchard that is 48 years old in Palestine by the Sea of Galilee. These trees are of the variety Majhul, and they are about 10 to 15 years tall. The height is usually appreciated by growers that it reduces disease incidence and protects the fruit from ground pests, but it also requires heavy machinery. The application of the ability to genotype commercial cultivars is essential and highly demanded within this industry. It allows the development into the level of future population and size. The agenda is as follows. As a soft caution, I will use the varieties and cultivars interchangeably throughout the presentation. Well known for its edible fruit, sugar, various polysaccharides, rich nutrient. It is a dioecious perennial monocot. Date palms are the major indigenous crop of the Arabian Peninsula. The date palm is one of the oldest cultivated trees in the world. Evidence of domestication dates back more than 5,000 years and extends further. The discovery of dates in the ancient pharaohs' tombs and the Neolithic sites around 7,000 to 8,000 years. Date palms are commercially propagated by the well-known method of tissue culture, micropropagation, mainly by two methods: somatic embryogenesis, which leads to major issues in mutation, and the industry standard that is recommended, which is organogenesis, which removes the need to go through the callus phase. This lengthens the period of initiation and the production prophase, as well as the overall length time required for successful multiplication. The genome consists of 18 chromosomes, pairs with 2n equals 36. There are known males that were distinguished by being polyploid, but no females have been observed. The size is roughly 670 megabase pairs. but it could be as large as 1 gigabase. We will know once the entire genome and its components are fully sequenced and published. Phoenix dactylifera, or the date palm, has been known to be successfully cultivated in the geographic area, in red mainly, in the Arabian Gulf, but it has been developed in the Americas, United States and Mexico, South Africa, and Australia. The geographic date palm distribution is quite dispersed, mainly above the equatorial line. We can clearly establish that the Middle Eastern countries, also known as the GCC nations, are leaders in this industry. The current global production exceeds an area harvested of 1.1 million hectares, or 2.7 million acres, and producing roughly 7.7 million metric tons. This is clear regarding the commercial significance of the date palm worldwide. There are around 3,000 known varieties, of which only 200 cultivars are propagated, and about 50 type products or byproducts from the fruit, covering industries of food, construction, products, and pharmaceuticals. Since the rationale of our research is based on the following. Since the 1870s, we have known about the Bayou disease, first established in Morocco, specifically in Marrakesh Oasis, which flourished thousands of years. This Bayou disease, a type of vascular wilt, is caused by fungus, Fusarium oxysporum forma albidennis specialis. Its main symptoms are the drying out, yellowing, and discoloration of one or more leaves in the middle crown. By the 1940s only, it had already devastated the date palm population. In 1 century, about 60% was lost, enabling the desertification sweeping in. We can see the amplitude of its wrath. The capability of disease epidemic, which is its spectacular trick, is persistent and longevity. It is well known as the common, considering the damage proportions. We must continue our hard work in preserving old plantations and producing clean stock that's true to type, genotypically clear and molecularly confirmed by Palmàprinting. Mixing, the other issue that we see in terms of the rationale of our research is the mixing of cultivars in large-scale production units, in tissue culture mainly, can take place, and the ability to fingerprint is essential to control the cultivars and the genotypes are pure. The other aspect is the climate change and the need for large plantations that are adapted to the future environment. For this research, the collections were made by myself and colleagues, requested by germplasm resources and sources, supplying fresh leaf materials that were made available across 7 countries, mainly comprising of the GCC countries, in which date palms grow readily, and due to the environmental correspondence between 2000 to 2013. The red dots on this map represent 28 different locations where 5 to 10 accessions for each of the 56 cultivars were collected. This was done in order to develop the first trilateral consensus in support of the cultivar identification fingerprint, also known as Palmàprinting. Historically, the indirect methods used to differentiate through proteins and phenotypes were used to infer genetic differences among individuals and populations, mainly cultivars. The direct methods today are the sequencing of few genes or selection of genes that are interesting. Today, the possible detection of variation among these individuals in cultivars through single nucleotide sites across the entire genome. The nice aspect of this is the non-active part of the genome, which was once known to be considered useless. Today we know that it holds far more value and usefulness in molecular assessment. For the most part, the relative popularity of microsatellites has increased since the 1995 onwards, with new technology such as SNPs, NGS, FLPs, perceived to go until perhaps 2025. But who knows? I think that we are to expect far more advanced technologies by then. For the purpose of this study and the need, no publication exists currently which exclusively provides specific consensus genotypes for the global commercial market of date palms. While new resultant techniques such as microsatellites, arrays, or microarrays, DNA chips provide cutting-edge, high-throughput solutions to date palm cultivar identification, they are expensive, involve highly specialized equipment, and require further characterization of single nucleotide polymorphisms, or SNPs. Therefore, more basic equipment or equipped molecular labs still utilize SSRs, or microsatellites, analysis for the purpose of cultivar identification, as we know today. Microsatellites occur within the introns and the exons. These are the regulatory regions, mainly in the non-coding part of the genome, or DNA. They present all eukaryotes and consist of several over hundreds of repeats between 1 and 6 nucleotide motifs. The locus specificity, in contrast to the multi-locus markers, or mini-satellites, or RAPDs, are not suitable, since they are distributed within the genome in such a way that recognizing them is challenging. Microsatellites are highly polymorphic, so this means they are hypervariable, and they provide considerable patterns that we can identify. The useful range that was also observed from individual identification to fine-scale phylogenies. According to the oligonucleotide primer sequence provided by Billiot and al. In 2004, we selected 14 specific primers that were selected in respect to the non-overlapping allelic ranges, the number of alleles detected on certain loci is also a parameter, and the heterozygosity. This allows us to create a routine panel and encompassing package where high confidence intervals exist in determining these genotypes. So the compatibility of primers was important for us due to the use of specific 5' modification amplification success to ensure proper typing of these successions collected, creating a consensus. So the 4 primers you see in panel 1 were sufficient for this study's goals, where microsatellite motif 10 and 35 were sufficient for the 6 FAM modification to be used for routine typing throughout the large-scale production by micropropagation on the field and in general verifications. The tailored system developed here for identification has proven itself for commercial and small-scale applications, including 4 microsatellites, low size 15, 25, 32, and 85. These microsatellites even now represent an easy, cost-effective way in identifying cultivars and varieties, and its applicability for commercial applications as well as laboratory setting has been facilitated. This is a sample of genotype data confirming the date palm varieties. The amplification products revealed that the amount of genotypic polymorphisms detected is sufficient to distinguish between 54 out of 56 of the world-renowned cultivars. Of the 56 cultivar population, 18 were non-confirmable. This suggests that there's much intravarietal diversity probably existing within the population. This could also correlate with the fact that selection much less is stringent on male varieties, which have fewer identifiable morphological characteristics. Whereas in female varieties are traditionally and strictly propagated through cloning or clonally propagated, in addition to being more easily identifiable by their various fruit characteristics. Further microsatellite markers will be required to encompass the remaining 18 out of the 56, but the results reveal that each were genetically distinct and unique due to their distances and relatedness as shown in the results. The other aspect that could be the cause of this is sympatric speciation. Sympatric speciation is the process through which new species or cultivars evolve or diverge from the single ancestral species while inhabiting the same geographic region. This is why, for example, the mijhouls, or one of the reasons why the mijhouls around the world phenotypically look and taste differently. It is true, it's a G by E interaction, so genetic by environment interaction, which causes these major phenotype differences, but ultimately it has also to do with perhaps sympatric speciation. These results suggest that the male cultivars and perhaps even some of the females, underwent a disruptive selection in its historical record. This is caused by either one gene or a linkage disequilibrium between genes of traits subjected to this disruptive selection, also known as ecotypes. Sympatric speciation was shown by distribution of pH and altitude being factors that cause perhaps intravarietal divergences and correlated by flowering phenology.
دكتور صلاح عشان الوقت بدنا نستعجل شوي لأنه
Yep. Going back to the results, within the 4 microsatellite panels, during fragment analysis, visual confirmation of the allele sizes on the electrophoregrams provided essential as the gene mapper software occasionally missed certain allele calls. Note the miscalled allele MDPCIR25 at 199 base pairs for the mijhoul due to the fragment being marginally outside the defined allelic range. This type of misrepresentation is non-conforming to the established consensus or the trilateral consensus we established. This has categorized these varieties as non-confirmable until additional microsatellites are tested.
Please conclude, Dr. Zaid.
Yeah, 30 seconds. Control samples of the 33 female and 5 males of different date palm cultivars. Additional sampling was done to triplicate to reach the consensus of these genotypes and to gauge whether any intravarietal variation is present. Luckily, the 5 males shown here, underscored in blue, we did not fall into this previously mentioned intravarietal divergence among them, and this was a good thing. These results enable the routine variety typing or Palmàprinting of in vitro cultures, greenhouses, nurseries, plants of point of sale, and on the field and beyond. So to conclude, the utilization of these microsatellite panel presents here enables the quality control monitoring of tissue culture and production in general, while assisting culture and plantlet management at any date palm micropropagation facility, plantation, or organization. With that, I would like to thank the Khalifa Award for the opportunity to share this valuable technology and knowledge and solutions to one of the industry's most challenging challenges in preserving and managing this valuable industry. Thank you for your attention. And if you have any questions, I'll take them now.
شكرا دكتور صلاح. يعني الصحيح أنا رغم أن الدكتور صلاح أخذ شوية وقت زيادة، لكن أنا على رأي I have a dream، والحلم اللي أنا موجود عندي هو أن أرى البنك الوراثي لنخيل التمر في الوطن العربي ويعمل على الأسس العلمية الدقيقة، وأعتقد أنها موضوع مهم جدا. في طبعا مبادرات طيبة في مواقع مختلفة، لكن أنا باعتقادي أن البنك الوراثي لنخيل التمر في الوطن العربي أصبح ضرورة ملحة، وأتمنى أن يتحقق هذا الحلم. وإحنا في أرينينا أيضا لدينا مجموعة خبراء وجاهزين أنه إذا تم هذا المشروع أن نساعد ونكون جزء من مسؤولية هذا المشروع. تفضل.